Why don't we jump into it today? A couple announcements before we get started. The next grand rounds is March 5th, and that's I'm going to butcher this name, Geriatric, with a topic to be decided. And our next journal club is March 12th via Zoom. And a couple other announcements following today's grand round. You'll receive an email with a link to the recording of today's presentation. Also included in the email will be an online evaluation. Your feedback is important to us and to the speaker, so please take a moment to fill this out, and please submit questions in the chat anytime during the presentation. So without further ado, I would like to introduce Roger Enoka, who completed his undergraduate training in physical education at the University of Otago in New Zealand. And prior to obtaining a master's degree in biomechanics and a PhD in kinesiology, both from the University of Washington, Seattle. He subsequently held a faculty position at the Department of Exercise and Sports Science and the Department of Physiology at the University of Arizona, which is in Tucson, and the Department of Biomedical Engineering at the Cleveland Clinic Foundation. He is currently a professor in the Department of Integrative Physiology at the University of Colorado Boulder, and he also holds joint appointments as professor in the Department of Medicine Geriatrics here and in the Department of Neurology here at the Anschutz Medical Campus. The overall goal of his research is to study the neuromuscular determinants of motor function in health and disease, and current themes of his lab include: one, the consequences of age-associated adaptations in neuromuscular properties on manual dexterity and walking performance, and two, the capacity of treatment with neuromuscular electrical stimulation to improve fatigue, fatigability, and mobility in persons with multiple sclerosis. So I would like to hand this over to Dr. Enoka, whose topic is fatigue and fatigability in health and disease.
Thank you very much for the introduction, and also for the invitation to be here. My first claim to fame is I survived the I-36 traffic to get here on time. But as mentioned, I'm going to talk to you about fatigue and fatigability. I began studying fatigue in 1981. The first 30 years of this work focused on the physiology of fatigue. We have written several reviews on this topic, and this one is the most highly cited of this work. Excuse me here. I figured it out, thank you. Thanks. So this paper has been cited 2,200 times. In 2012, I was invited to co-author a paper with Benzie Kruger in the Department of Neurology at Anschutz. She's here we go. Excuse me, working. I'm trying to move this here. Oh, okay. There you go. Thank you. So as I said, in 2012, I was invited to co-author a paper with Benzie Kruger in the Department of Neurology at Anschutz on the topic of fatigue and fatigability. This was a significant experience for me, and today's talk is based on that collaboration. So there are many stakeholders with an interest in fatigue, but the problem is that we do not speak the same language. For example, fatigue means one thing to somebody, to a clinician, but something quite different to a physiologist. The objective of today's talk is to describe a comprehensive framework that can enable us to talk to each other about fatigue with a common language. To achieve this objective, I have organized the talk into 4 parts. The first part is going to be on some clinical observations. First of all, one study on older adults and then one on multiple sclerosis. Our interest in fatigue stems from its negative impact on performance. One way to frame the issue is to base the discussion on clinical observations. In the Chianti study, enrolled 1,155 adults aged between 65 and 102 years to examine the association between fatigue and physical function. They were asked questions about fatigue based on recollections during the preceding week. There were 4 choices for each question. The first choice was less than 1 day per week. The second was 1 to 2 days per week. The third was 3 to 4 days per week. And number 4 was 5 to 7 days per week. Respondents who selected more than 3 days per week were classified as fatigued. Participants who reported being fatigued in the preceding week, 15.3% of them were men and 29.1% of them were women. There were, there's a mix up here. The participants were assessed based on clinical conditions, 10 comorbid diseases such as hypertension, coronary heart disease, congestive heart failure. Still given. Slowly getting it. Sorry, diabetes, chronic bronchitis, hip fracture, cancer, arthritis, cognitive function, and depression. And the biomarkers were hemoglobin level, C-reactive protein, IL-6, serum tumor necrosis factor alpha, and thyroid function. However, there were no significant differences in either the clinical conditions or the biomarkers. However, there were significant differences between groups in motor function. These findings are about older adults who self-reported symptoms that were interpreted as fatigue. The results were similar for males and females. Hand grip strength was weaker in the fatigue groups for both males and females, and the fatigue participants reported more disabilities as estimated with the instrumental activities of daily living instrument. The fatigue participants had a lower average speed when walking 400 meters as a test of walking endurance. There was a greater percentage of them were unable to walk 400 meters. So now on to an example from multiple sclerosis. As you know, it is an immune disease that damages the nervous system, that causes nerve cells to die and impairs the transmission of activation signals between nerve cells and muscle. The graph plots the changes in gray matter fraction for the whole brain over 18 months as assessed by MRI. This provides a crude index of the number of neurons in the brain. People diagnosed with MS experienced a greater decline in the amount of gray matter atrophy compared with the control group. The key symptoms for people with MS include elevated fatigue, difficulty with walking, declines in daily levels of physical activity, and reduction in the quality of life. Approximately 80% of individuals living with MS list fatigue as their most debilitating symptom. Fatigue in MS and other neurological diseases is often quantified with questionnaires. The fatigue severity scale is one such questionnaire. Respondents are asked to rate 9 items listed on the slide on a scale of 1 to 7. ranging from disagree to agree. An average score of more than 4 across the 9 items is interpreted as a clinically significant level of fatigue. So based on these kinds of findings from older adults and individuals with neurological disease, we consider fatigue to be a disabling symptom. It represents feelings of tiredness, lack of energy, low motivation, and difficulty in concentrating. It can only be measured by self-report. And fatigue compromises physical and cognitive work capacity, which refers to fatigability. Note the distinction here between fatigue as a symptom and fatigability as a measure of work capacity. Let us return to this questionnaire, the FSS scale, and consider items 2, 6, and 9. These items indicate that the assessment of the level of fatigue for a person with MS can be estimated from questions about its impact on functional capacity, that is, fatigability. Conversely, perceived levels of fatigability reflect the fatigue status of the person. So questionnaire responses are based on recollections of challenges to homeostasis and depending on the psychological state of the individual. So the adjustments that occur that influence homeostasis include changes or alterations in any of these variables listed from blood glucose through to wakefulness. These adjustments are perceived and are used to estimate the current level of increase of fatigability. For an example, an increase in the level of circulating cytokines, as occurs in many diseases, can elevate the perceived level of fatigability. Similarly, the perceived level of fatigability is influenced by the psychological variables included in this list. For example, perceived fatigability is elevated during depression and reduced by reward. Because fatigability refers to the influence of the symptom of fatigue on work capacity, perceived estimates of fatigability are related to the level of fatigue reported by an individual. So now I will move on to the second part of the talk, and I will briefly discuss some of the findings on the physiology of fatigue. Most research on fatigue over the last 125 years began with the work of Mosso. He was a professor of physiology at the University of Turin and published a book on fatigue in 1891. Mosso proposed that fatigue emerges from 2 sets of adjustments, those that influence contractile function and those associated with the sensory features of physical activity. Most studies of fatigue performed by physiologists, however, have examined adjustments in work capacity, that is, fatigability, and not the symptom of fatigue, and hence the disconnect between work of clinicians and those of physiologists. Nonetheless, the involvement of muscular and sensory components, as proposed by Mosso, has persisted in this field. Physical activity involves 2 sets of physiological processes, those required to generate an activation signal, as indicated on the left, and now those that control contractile function, shown on the right. All voluntary actions begin with the generation of a motor command. In the primary motor cortex and its transformation into the activation of the thick and thin filaments in muscle fibers. Impairment of any of the processes shown in this diagram can influence fatigability. This means that there is no single cause or explanation for declines in work capacity. Instead, the adjustments depend on which of these processes is most stressed by the activity that is being performed. One experimental approach used to study fatigability is to measure changes in an applied force and the level of muscle activation during a prescribed task. This figure shows a person applying an upward force with the wrist by contracting the elbow flexor muscles. Gradual increases in the applied force, shown by the green trace, are produced by a progressive increase in the activation of the elbow flexor muscles. Muscle activation is measured as an electromyogram, as shown by the red traces. The EMG recordings show increases in the activity of the short and long heads of biceps brachii and the antagonist muscle, triceps brachii, during the increase in the applied force. This experiment shows a series of submaximal isometric contractions performed with the elbow flexor muscles for as long as possible. The target force for each 6-second contraction was 50% of maximum. Once each minute during the task, the participant performed a maximal voluntary contraction indicated with an arrow as an MVC. The bottom trace shows the progressive decline in MVC force across the protocol achieved during the series of intermittent contractions. At the same time, there is a gradual increase in EMG amplitude for the 4 elbow flexor muscles. This figure shows that the task decreased force capacity, as indicated by the decline in MVC force, and required an increase in the amount of muscle activation to achieve the target force. This graph plots the MVC force measured during the experiment shown on the preceding slide. It shows the rate of decrease in MVC force during the series of intermittent contractions. The time at which the applied force last reaches the target force is known as the time to failure. This is a measure of fatigability. Similarly, the slope of the line provides another measure of fatigability. As shown on this slide, the most common approach used by physiologists to measure fatigue involves the decline in the MVC force, the maximal force, after performing a prescribed bout of exercise. However, this measurement cannot indicate the level of fatigue because it does not reflect a self-report of symptom intensity. Instead, it provides a measure of fatigability. One approach used to determine the relative contributions of muscle activation and contractile function to declines in force capacity is to compare adjustments in voluntary and electrically evoked muscle contractions. Voluntary contractions arise from motor commands in the brain and involve all the processes from the motor cortex down to the contractile proteins. In contrast, electrically evoked contractions begin in nerve branches within the muscle, thus bypassing most of the nervous system. This slide shows a protocol used to compare adjustments in force during voluntary and electrically evoked contractions. The task was to perform a series of intermittent contractions with the elbow flexor muscles. The target force was 50% of maximum. Intermittent contractions were 6 seconds of contractions followed by 4 seconds of rest. Once every minute, the participant performed a maximum voluntary contraction to determine force capacity during a voluntary contraction. In addition, the muscles were stimulated with electric shocks, either as single shocks or as a series of shocks. The single shocks elicited twitch responses as shown here. The series of shocks, shown as 50 Hertz, provides a measure of the true force capacity of a muscle. This slide compares the decreases in voluntary and electrically evoked force during a series of intermittent contractions. The target force, again, was 50% of maximum. The voluntary force is shown with black symbols, and the force evoked with stimulation at 50 Hertz is shown with gray symbols. The graph shows the results for the knee extensor muscles. There was no difference in the decreases in the voluntary and the electrically evoked force during this task. This means that the activation signal generated by the nervous system was able to elicit the maximal capacity of the muscle. In contrast, the graph on the right shows that the voluntary force for soleus declined more quickly than the electrically evoked force. At task failure here, for the voluntary contraction, the force elicited by electrical stimulation was 70%. So this difference, it represents a deficit in voluntary activation. For this muscle, during this task, the nervous system was unable to provide a sufficient activation signal. So deficits in muscle activation are quantified with a measurement known as voluntary activation. The graph shows the force generated by the dorsiflexor muscles of a 50-year-old man with ALS. The first part of the trace over here shows the peak force he produced during a maximum voluntary contraction. When electrical stimulation was applied, there was an increase in the measured force. This means that his nervous system was unable to provide an activation signal that could elicit the true force capacity of the muscle. This deficit is quantified as the ratio of these two forces, and it's known as the voluntary activation. And what this number means is that for this person, during this task, his nervous system was only able to evoke from the muscle 74% of its true force capacity. This graph compares the declines in MVC force on the left axis and voluntary activation on the y-axis for a task that lasted 1.5 minutes. And the task was to maintain maximal voluntary contraction for this duration. And what you see is after 1.5 minutes, the MVC force declined to 30% of its initial value. In contrast, the level of voluntary activation had declined by only 20%. So this comparison means that the decline in MVC force was mainly due to adjustments that occurred in the muscle and not in the nervous system. So during strong contractions, the capacity of a person to perform a contraction depends on adjustments that arise related to contractile function. Now, in contrast, when a person performs a weak contraction, in this case 15% of maximum for 43 minutes, at the end of the task, MVC force was 60% of the initial value, whereas voluntary activation was 70%. So this comparison here means that this decline in voluntary force was largely explained by reductions in voluntary activation. During weak contractions, therefore, most of the fatigability is due to impairments that arise in the nervous system and not due to problems in the muscle. So based on responses to questionnaires, I have described how self-reports about the level of fatigue can be derived from perceptions about challenges to homeostasis and the psychological state of the individuals. These perceptions are derived by questionnaires that ask about recollections during the preceding week. The perceptions provide estimates of the current level of fatigability, that is, a potential to do work. We can add to this framework adjustments in contractile function and voluntary activation during the performance of the task. These types of impairments contribute to adjustments in measured fatigability as distinct from perceived or estimated fatigability. Known factors that contribute to declines in function include alterations in these factors listed here, whereas alterations that depress voluntary activation include this list. So both estimated and measured fatigability reflect a decline in work capacity, either or both cognitive and physical work capacity. Estimated fatigability reflects decreases in the capacity to perform past and future actions. And I will explain what I mean by future in a moment. In contrast, measured fatigability reflects limits on current actions. For example, the capacity to sustain a task depends on the rates of decline in contractile function and the level of voluntary activation. Outcomes that can be used to measure fatigability include this list here, such as the decline in the maximum voluntary contraction force, time to task failure, rating of perceived exertion, and the Stroop test. Instruments that can be used to estimate fatigability include a fatigue severity scale, as I indicated, and other similar scales, and whereas the bottom two items, rating of perceived exertion and the visual analog scale, can be used to obtain instantaneous estimates of fatigue. So now to the unifying framework based on this background. In this talk, I have defined fatigue as a symptom that reflects feelings of tiredness, lack of energy, low motivation, and difficulty concentrating. Fatigue compromises work capacity, either physical or cognitive work, in which we estimate with measures of fatigability. Estimates of fatigability are modulated by challenges to homeostasis and the psychological state of the individual. These estimates reflect decreases in the capacity to perform past and future actions. Measured fatigability is influenced by the decline in contractile function and voluntary activation, and these adjustments limit the capacity to perform current actions. Feedback from sensory receptors during the performance of a task will alter the perceptions that are used to estimate fatigability, the level of fatigue, and voluntary activation and contractile function. So now to the last part of the talk, translating this framework to practice. And I will discuss two examples, one related to multiple sclerosis and the other related to something known as pacing. Fatigue in MS can be estimated with this questionnaire, as I mentioned. So respondents are asked to respond to these 9 items on a scale of 1 to 7, and an average score of greater than 4 is interpreted as a clinically significant level of fatigue. In the study that I'm going to describe, the MS group, the FSS score was 5.3, meaning on average clinically significant fatigue. So the purpose of the study was to explain the variability in the self-reported level of fatigue in a group of individuals living with MS. So this is the questionnaire results. So the study was performed on a hand muscle because it was assumed that this was less deconditioned by the sedentary lifestyle of most people with MS. Participants included 20 individuals with MS and 20 sex and age-matched controls. The force generated by a hand muscle was measured as an upward abduction force applied by the extended index finger. Strength was measured as the MVC force, maximum voluntary contraction force. When normalized as a Z-score compared to the control group, this was a significant reduction in strength for the MS participants. The task was to sustain an upward force at maximum for 2 minutes. And fatigability was measured as the rate of decline in the sustained force over the 2-minute period. The fatigability did not differ between these 2 groups, the MS group and the control group. They used these data to explain differences in the self-reported level of fatigue, that is the fatigue severity score, for the MS group, and the regression equation they derived explains 77% of the variance in that fatigue score. So the most significant predictor was the decline in force, that is the measure of fatigability. The second was the strength of the muscle, MVC force, and the third was depression. So if we go back to this scale here, the framework, the predictive variables were measured fatigability, the strength of the muscle was indicated by the combined action of contractile function and voluntary activation, and the level of depression reported by these individuals. In a follow-up study, the same group of investigators compared adjustments during a 2-minute MVC. So here's the 2-minute MVC. They applied electric shocks during the MVC, and each shock evoked a twitch force, as shown here, and the evoked forces were used to derive a measure of voluntary activation, as I've mentioned. The graph compares the decline in force during the sustained MVC for the control group and the group of MS patients. And as reported, there was no difference in fatigability. That is, the capacity of this hand muscle to sustain this force did not differ between these 2 groups of individuals. However, there was a significant difference in the rate of decline in voluntary activation. So the rate of decline in force, to remind you, represents muscle function, contractile function, and voluntary activation. There was no difference in that, but there was a difference in the activation signal. So when they explained the decline in force, they found different predictive variables. So for the patients, 51% of their fatigability was explained by the decline in voluntary activation, whereas for the control group, 49% was explained by the difference in the strength of the hand muscle. So although fatigability was the same, the underlying predictors differed for the 2 groups. Now I will discuss the second example, which is known as pacing. This example has to do with the influence of feedback from the ongoing action on the interaction between estimated fatigability and measured fatigability. The study compared the influence of changes in brain function on time trial performance. So 10 athletes were recruited for the study and performed a crossover design with 3 drug conditions, placebo and reuptake inhibitors for noradrenaline or dopamine. Protocol included 3 outcome measures, TMS, transcranial magnetic stimulation, to measure responsiveness of descending pathway. This is ES here, electrical stimulation of the femoral nerve to assess voluntary activation, and PVT, which is a psychomotor vigilance test. It's a reaction time test to visual stimuli. So the time trial was to cycle for 30 minutes at 75% of maximal power output as quickly as possible. After the cycling exercise, participants performed the same 3 tests, TMS, electrical stimulation, and the vigilance test. Here's a comparison of the power applied to the pedals under these 3 drug conditions: a placebo, depression of the motor pathways, and depression of the arousal pathways. And what the graph shows, these are the same individuals. They were, of course, blinded to the drug condition. And you can see there was a marked depression or decrease in the power applied to the pedals under this arousal depression condition, but no difference between the other 2 conditions. So the time to complete the time trial did not differ between the placebo and the motor condition, but it was longer under the aroused condition, which means that the participants exhibited greater fatigability when the reuptake inhibitor that influenced the arousal pathway was depressed. Similarly, the psychomotor vigilance test the reaction time was longer, meaning that the reaction was impaired under this condition. During the task, they had participants answer this rating of perceived exertion, the classic scale from 6 to 20, and they were asked to rate at this instant in time, what is the relative perceived exertion? At task failure, what you see is there was no difference in the RPE value. There was a decrease in the power applied to the pedals. There was a change in psychomotor, but no difference in perceived exertion. These findings of these studies indicate that measured fatigability was greater when arousal was reduced, but there were no differences in the perceptions underlying estimated fatigability. And the conclusion of the study was that adjustments in the processes that influence measured fatigability, that is, the power applied to the pedals, were regulated by perceptions of the challenge to homeostasis. This means that future actions regulate the my arrow keeps disappearing, sorry. This regulation is referred to as pacing, and this involves maintaining the maximal effort possible while not compromising the goal of the task. In time trials, this means covering the specified distance as quickly as possible. So here are the key points I've attempted to make today. First of all, fatigue is a disabling symptom. It reflects feelings of tiredness, lack of energy, low motivation, and difficulty in concentration. It can only be measured by self-report. Fatigue compromises physical and cognitive work capacity, which we refer to as fatigability. And the factors that establish the level of fatigability vary across individuals and conditions. If you get nothing else out of my talk today, be careful how you use the F word. Thank you.
Thank you, Doctor. Thank you, Dr. Enoka. Wonderful talk. While individuals in the audience are thinking of questions, I'll start with one. I think of an older person who is fatigued, who has fatigue. And I wonder, and we could produce fatigability in that older person by having them exercise, for example, or having them participate in physical activity. What do we know about inducing fatigability in someone who is fatigued? Does that worsen fatigue? Does that improve fatigue? Do we know anything about that? How those two interact?
So we know that the level of fatigue can be estimated by self-report. And this self-report value compromises work capacity, meaning the greater the level of fatigue, the intensity of the symptom, the greater the reduction in work capacity. Does that answer your question or not quite?
Well, that goes to, I think, how fatigue could influence fatigability. I'm curious about the reverse. How does fatigability, or the presence of fatigability, does that influence the self-reported fatigue?
So, to a certain extent, this literature is kind of circular in nature, because, as I mentioned early on, to estimate the level of fatigue in an individual, healthy or otherwise, we use questionnaires, and some of the items on the questionnaire are asking about fatigability. So to that extent, it's difficult to disentangle the two.
Other questions?
Hi, Dr. Enoka. Thank you for that talk. I found this actually fascinating. I'm thinking from a clinical perspective of a geriatrician. I practice mostly in an inpatient setting. And, you know, a scourge to us is just the problems of deconditioning when people stay in the hospital, either from the health condition or the immobility experience. And also plenty of questions with regards to reconditioning. What's the likelihood that someone's going to recover or not recover after one of these events? And what I took from this talk was that you have several different measures to examine different patterns and how people's muscles and or neurology impacts their ability to use those muscles. My question was from a practical standpoint, like the technical measurement of these patterns, like how small are the devices that you use to do this? Are these devices that can be taken out of the lab and go into the setting and measure some of these things? Or are these devices that are like very, very large, hard to set up and to get good measurements? I was curious to know a little bit more from your experience.
So, as I indicated, there are several ways to measure fatigability. There are a number of instruments that I use depending on the cohort to estimate the intensity of fatigue. But there are many methods that can be used to estimate fatigability. So, for example, one that we and others use for people within our MS studies, we have them do a test of walking endurance. So there are many versions of this. A common one is how far can you walk in 6 minutes when you're walking briskly. So what you can do is take the distance covered in the first minute relative to the distance in the 6th minute, and that provides a measure of fatigability. So there's no devices involved at all. And so there are some very simple tests that can be used.
Thanks.
That was a very interesting talk. I was thinking about the graph you showed between the strong force versus the weak force and the differences in the stimulated or the contractile force and the voluntary force. And my thought was the differences in the motor recruitment between large and smaller motor recruitment. And then the devices that you use clinically, I think you showed was in the hand. And so I'm curious if you have different devices for different muscle types. So different recruitments for the smaller muscles in your hands versus perhaps like the soleus you mentioned and even the quad to show differences in neuromuscular dysfunction.
Fundamentally, the devices are similar for all kinds of muscle groups. The graph that you're referring to, I didn't mention it, was actually performed on the elbow flexor muscles. And so that same finding has been reported for leg muscles, hand muscles. So it's a common finding. One point I didn't mention, which I would like to now, is that distinction between the adjustments that limit performance during strong contractions versus weak contractions. It's muscle focus in the strong one and nervous system in that weak one. And what I would like you to interpret from that is most of the fatigability that you experience during activities of daily living is mainly due to adjustments that occur in the nervous system and not in the muscle.
I'll follow up on that. So as individuals age, we know there's a fiber type transition from, in some muscles, from slower to faster fiber types that are thought to be more fatigable. Do we see the pattern switch as the muscle fiber types become faster as individuals age?
So what we know about, so in muscles, classic studies, muscle physiologists identify 3 types of muscle fibers depending on the type of technique that you use. And the proportion changes as we age. So there's a decrease in the large, fast muscle fibers. But these adaptations seem to have little influence on fatigability.
Other questions?
We're constantly trying to tease out in the clinic, and so this is a fatigue, is the contribution of sleep deprivation or lack of sleep. I'm curious, I know this wasn't a part of the topic, but if there's any work out there on how sleep deprivation impacts some of the measured fatiguability. You see, like, what effect does that have if you were to intentionally deprive subjects of sleep?
You know, I'm not so familiar with that literature. I do know that in one of the early slides, which I showed the variables that can influence estimates of fatigability, one of them is wakefulness. And there are for sure that as wakefulness declines, the estimated fatigability increases. So I assume that with sleep deprivation, like strange working hours, for example, contributes to that as well.
Perfect. Thanks for a great talk. I was curious from an intervention perspective in the context of latency age, from your perspective, do you think there's any, or what would you recommend as a potential intervention for mitigating fatigability in individuals as they age? I'm curious from a physiological perspective, and I think this would also be relevant for clinicians.
So what I've done today is kind of laid out a framework that how these things might tie together that enable clinicians and physiologists to talk to each other in a common language. That's the main point. But your question raises a very important point. We do not, if people are willing to accept the framework, we do not know the details yet. So first of all, we need a buy-in on that framework, and then we need investigations that probe the boundaries and the influence, the interaction between the different variables. So the field is very weak in providing that kind of an answer. And again, related to your question, the fatigability that you might measure in an individual will depend critically on the task that you use. So if you're interested in clinical outcomes, then you have to use a clinically relevant task to get an ecologically valid measure of fatigability.
Thank you.
Other questions? Okay, with that, I'd like to thank Dr. Enoka for a wonderful talk.